Hello everyone. In this video we'll be talking about type 1 hypersensitivity. Now, as we all know, there are four main different types of hypersensitivities, and they are going to be explained in further videos that are going to be uploaded on this channel later on. So let's talk about type 1 hypersensitivity. Essentially, we've got an allergen that is going to be entering into our body, and as it is detected, it is going to be cleaved and it is going to be presented to plasma cells by the help of the T helper cells. Recall that the T helper cells are going to be facilitating, they're antigen presenting cells, so they're going to be getting these small pieces of proteins that are present from these allergens, and they are going to be incorporating them as if, let's imagine I am the antigen presenting cell, in this case the T helper cell, and then I'm this hand, this arm is going to be the MHC, and I'm going to be holding a piece of the allergen. So, and in this case it is called an antigen. So basically what's going to be happening here is that a plasma cell is going to be detecting this antigen, and it is going to be producing, like all B cells do and plasma cells do, they're going to be producing antibodies against this antigen. So what is happening here is that now we've got antibodies against this specific antigen. So in essence, these antibodies are going to be of the type IgEs. Now these IgEs are going to be binding. Now this is the catch. These IgEs are going to be binding to the Fc epsilon receptor 1 that is present on the mast cells. When this happens, these mast cells are going to be sensitized. We mean by that these mast cells are now able to get triggered by the action of allergens. Let's actually figure out how. Essentially, let's imagine we've got, just like we mentioned before, we've got these IgEs binding to the Fc epsilon receptor 1 that are present on the mast cells. So let's imagine there are two of these receptors and these IgEs bind to them. What is happening here is that one allergen is going to be binding between two IgEs, and after this happens, we're going to be these mast cells are going to be releasing mediators. Now let's understand what these mediators do. So there are two main phases that are going to be happening because of the release of these mediators. First of all, we've got the histamine. Essentially, the histamine is going to be binding to the H1 receptors that are present on the surface of cells on the respiratory airways, and these will trigger symptoms of allergies. Now, this is quite one of the more common examples, so this is pretty much applicable in our daily lives. Next, we've got the delayed or the late phase that is going to be happening due to the release of these mediators by the mast cells. Essentially, we know that there are membrane phospholipids, and they're going to be released, and they are going to be giving prostaglandins, like for example the D2, or leukotrienes like the B4, the C4, and the D4. Now these are going to be acting as chemokines. Recall that the chemokines are chemical signals. So they are going to be releasing chemokines that are going to be attracting leukocytes to, or white blood cells, to the site of infection, and this is going to be inducing epithelial damage and bronchospasms. I hope you enjoyed and gained something new from this video. I hope you drop a like down below and subscribe. Thank you for watching.